Hello everyone, welcome to Raaonline. Today we will discuss our new chapter, that is biotechnology and its application. In previous we'll discuss about the processing, right? We discuss about the processes and in that chapter how we apply it, right? So in biotechnology and its application we will study about the therapeutics, the diagnostic, the genetically modifying crops for the agricultures, right? For processed food, the medicines, genetically engineered medicines, right? All these things and the sources for the energy production, right? All these things we'll study under the biotechnology, right? Come to the first question. We'll study the whole chapter in the form of the questions, right? First, select the incorrect statement related with the Green Revolution. Right? It is also given in NCERT, that is incorrect statement. We have to choose the incorrect statement, right? So, succeeded in quadrupling of the food supply? No, it is related with the tripling of food supply, right? That is mentioned in NCERT exactly. Increase was insufficient to feed the growing human population? Yes. Increase the yields could be contributed to improved crop varieties? Exactly. The use of agrochemicals contributed to enhance the yields? Exactly. So in terms of incorrect, the answer is the first, right? Now we will have to study about that, what is green revolution. So come to here. See, this is the exact lines of the NCRT, and in NCRT it is mentioned that the green revolution succeeded in tripling, not the quadrupling, right? That is tripling the food supply, but yet it was not enough to feed the growing human population, right? As per day the human population increases per day very fast, right? So according to that, it was not sufficient to provide the food everyone, right? To provide the necessity, to complete the necessity of everyone. So increased yields have partly been due to the use of improved crops varieties, right? But mainly due to the use of better management practices and the use of the agrochemicals. But remember one things, right? These are the fertilizers and the pesticides, right? Agrochemicals, that is the fertilizers and the pesticides. But for the farmers in the developing world, agrochemicals are often too expensive to use, right? And for further increase in yield with existing varieties are not possible, so you by using the conventional breeding, right? So that's why all these lines all these statements are the correct, except that because succeeded in the tripling of the food supply, it's not quadrupling, right? That is related with the green revolution, and green revolution is related with the agriculture, right? Come to the next question. Methods of producing microbes and pest resistant plant include microbes, that is disease causing organisms, right? These are the organisms, microorganisms. And pest resistance plants include, what will happen, the RNAi. What is that RNAi? That is related with the that is the method for producing the microbe and pest resistant. What is RANI? That is the RNA interference, right? That is the RNA interference, right? Use of the Bt toxin, correct, right? Pest resistant plants, right? Gene therapy? No, gene therapy is not related with that. So the answer is both 1 and 2, right? How? See that, what is RNAi? That is RNA interference is a biological process, right? That RNA interference is a biological processes in which the RNA molecules inhibit the gene expression, right? In which these RNA molecules inhibit the gene expression or the translation by neutralizing the targeted mRNA molecules, right? By neutralizing the targeted mRNA, that is the messenger RNA molecules, right? So that RNA interference is the biological process. When we talk about the BT, see here, that is we say that the use of BT toxin. What is that BT? That BT stands for the Bacillus thuringiensis, right? That Bacillus thuringiensis is a species of the bacteria that produces the proteins that are toxic for certain insects, right? That are toxin for certain insects. Because of this, it has been used as a safe microbial insecticide, right? It has been used as a safe microbial insecticide for over 50 years to control the pest caterpillars, right? Clear? To control the pest caterpillars. That BT toxin, that BT, that is Bacillus thuringiensis, makes that toxin which target to the insects larva when eaten, right? And what will happen in their gut? That toxin are activated, right? And what will happen? That activated toxin breaks their gut wall and insects die of infection and the starvation, right? So death can occur within few hours or few weeks, right? So that's why in that manner, that pest resistant plant, by the use of the BT toxin, that is Bacillus thuringiensis bacteria, release a toxin which kill the insect, right? Now come to the next question. Which one of the following is not an advantage of the GM crop, right? What is that GM crop? That is genetically modifying crops. Genetically modifying crops, right? Genetically modifying crops are called the GM crops. Actually, what is not an advantage in that question? It is talking about the not an advantage, right? So when we talk about the advantages, so these advantages are the, it increase the crop tolerance to salt in the soils, right? It reduces the post harvest losses, it increases the efficiency of the mineral uses, right? And decrease the reliance of the chemical pesticides, right? So the answer according to that question, the answer is 2, that is increased reliance of the chemical pesticide, right? Now come to here, that is what is genetically modifying organisms and the what are the crops, right? These are the exact line of the NCERT, and you have to focus on these lines so you can solve all these the questions easily, right? See, in NCERT it is clearly mentioned that the plants, the bacteria, fungus, the animals whose genes have been altered by the manipulation, right, they are called the genetically modified organisms, right? They are genetically modified organisms, that is GMO, right? That genetically modified organisms, that is GMO. These GM plants, genetically modified plants, have been useful in many ways. How? That genetic modification has made crops more tolerant to the antibiotic stresses, right? That is the cold, drought, salt, heat. So according to that, that statement is correct. That is the advantage of that, right? Second, reduce the reliance of chemical pesticide, that is pest resistant crops, right? And in second, that option, we use the increased, not the decreased. So that statement is wrong in that way, right? Clearly in NCERT it is mentioned that reduced reliance on the chemical pesticides, right? Third, it helped to reduce the post harvest losses, right? That's why that statement is correct. Third, the statement is also taken from that, right? Increase the efficiency of mineral uses by the plants, which prevents the early exhaustion of the fertility of the soil. Exactly, that is also the fourth statement. That's why these three statements are correct and this one is incorrect. So you have to know these statements already, right? And the last one, it enhances the nutritional value of the food, right? Because it is genetically modified, so the traits which is necessary to for the benefit of the for the beneficial for the human races, right? That the vitamin A enriched rice, right? So in that manner, these genetically modified crops enhance the nutritional value of the food, right? That is vitamin A enriched rice for the example. So these are the advantages of the GM crops. Clear? Come to the next question. Production of pest resistance plants could first increase the amount of pesticide used, increase the amount of weedicide used, decrease the amount of pesticide used, both 2 and 3, right? Now, according to that, the production of pest resistance plant, why to decrease the amount of the pesticide used? If we produce the pest resistant plant, then what will happen? There is no need to apply more pesticides, and to prevent them, we prevent indirectly ourselves, right? Because when we eat these pesticide used crops, right, maximum amount of pesticide used crops, then it will harm to our body also, right? So in NCERT, exact that line is given in page number 208 12.1 heading, right? That is the production of pest resistant plant which could decrease the amount of pesticide used, right? So the answer is 3. Identify the gene, its protein that controls the corn borer from the given option, right? Corn borer and cotton bollworm, there are two. So the corn borer is controlled by that cry1AB and cry1AB, that is the cry gene, right? That is the cry gene, that is related with the cry gene. And what is that? So come here. Bacillus thuringiensis produces a specific toxic genes which were isolated from that Bacillus thuringiensis, right? And these toxin genes incorporated into the several crop plants such as the cotton, right? Such as the cotton. And the choice of genes depends upon the crop, right? That choice of genes depends upon the crop and targeted pest, right? So as most Bt toxins are the insect group specific. These Bt toxins are the insect group specific. So these toxins are coded by a gene which is called the cry gene, right? These are called the cry gene, and there are a number of them. For example, the proteins which are encoded by the genes, that is cry1AC, cry2AB, and both of them control the cotton bollworms, right? They control the cotton bollworms. And that of cry1AB, it controls the corn borer. It is the exact line of NCERT. From that line that question arises, right? So you have to understand the exact lines of the NCERT, what will happen with that line, and how many informations are condensed in that particular one line, right? So you can solve easily all the questions related with that, right? Come to the next one. Crystalline protein synthesized by the Bacillus thuringiensis, it is activated by. What will happen in that case? As we already studied that Bt toxin, when that Bt toxin is inactivated in state, right? But when the insect ingests it, it gets activated, and what will happen? It perforate its gut wall, right? And in that manner, what will happen? The insect is died, right? So what will happen? It is activated by the acidic condition of the bacterial food vacuole. No. Alkaline pH, yes, alkaline pH, but of the bacterial food vacuole, no. Alkaline pH in insect foregut, no. that is alkaline pH in insects midgut. Exactly, that is the exact line of NCERT. See here, actually that Bt toxin protein exists as inactive prototoxin, right? But once an insect ingests the inactive toxin, what will happen? It is converted into active form of toxin due to alkaline pH of the gut, right? Here he just used the word gut, but in that particular portion he used the surface of the midgut epithelial cells, right? So you have to learn that the alkaline pH in insects midgut, right? The crystalline protein synthesized by the Bacillus thuringiensis is activated by the alkaline pH of the insect's midgut wall, right? The activated toxin binds to the surface of that midgut epithelial cells and creates the pore that causes the cell swelling, right? And the lysis, and what will happen eventually, it is the cause of the insects, right? So in that manner, that Bt toxin acts as a insecticide, right? Come to the next question, that human insulin is being commercially produced from a transgenic species, right? What is that transgenic species? So first we have to know about that, right? See that is the Saccharomyces E. coli, Mycobacterium or Agarobacterium. So the answer is E. coli. What will happen? In 1983, Eli Lilly, an American company, right? They prepared two DNA sequences, right? They prepared two DNA sequences corresponding to A and B, right? A and B chain of the human insulin. As we know that we already discussed in the previous chapters, right? The insulin is the 51 amino acids, has the 51 amino acids which are arranged in two chains, that is acidic A chain, basic B chain, right? And what will happen? Introduce them in plasmids of the E. coli, that is Escherichia coli, to produce the insulin chains, right? So that chain A and B were produced separately, extracted and combined by creating the disulfide bond to form the human insulin, right? To form the human insulin artificially, right? So in that manner, that human insulin is commercially produced by the transgenic species of the Escherichia coli, right? Come to the next question. Bt toxin kills the insects by, we already studied that Bt toxin kills the insect by creating the pores in the midgut wall, right? Blocking mitochondrial respiration, no. Blocking transfer of nerve impulses, no. Creating pores in the body surfaces, yes, but at what surface? Creating pores in midgut epithelial cells, not the outer body surface and all these things, right? So it creates the pores in midgut epithelial cells. We already discussed in previous question, so the answer is 4. See that actually the Bt toxin that protein, we already discussed it in previous question, right? So that is the explanation of that question. Clear? Come to the next. How many recombinant therapeutics have been approved for the human uses in the world over, right? This is the exact line of NCERT also, the in NCERT page number 210, right? Heading 12.2, it is given that at present about 30 recombinant therapeutics have been approved for the human uses all over the world, right? But when we talk about the India, in India 12 of these are presently being marketed, right? So in that case the answer is the Bombay, right? Come to the next question. Which one of the following may be considered as an application of the biotechnology? That is bioremediation, waste treatment, energy production, all of these. So the answer is all of these, right? So the answer is the all of these. Why? See, the question is, that is also the line of NCRT which is given in that, right? It may be considered as the application, right? Application of the biotechnology. So see here, what is that bioremediation? That is the branch of the biotechnology that employs the use of the living organisms, right? Bios, that is living organism, is involved in it, and we use to treat something, right? That's why it acts as a remediation. So these microorganisms are like the microbes, the bacteria, right? In the removal of the contaminants, the pollutants, and the toxins from the soil, the water, and the other environments, right? So the bioremediation is used to clean up oil spills and contaminated groundwater, right? Now come to the next method, that is waste treatment. These methods are based on the biotechnology in waste water treatment. Actually, they are activated the sludge, the trickling, the filters, the oxidation ponds, the biofilters, and the anaerobic treatment. Right? What will happen? Solid waste compositing techniques, the biotricking filters, and the bioabsorption are the examples of the biotechnology applications which is used in the environmental engineering, right? Now come to the third, that is energy production. The contributes of that biotechnology for the energy industry, they are not restricted to the production of the biofuels, right? Or the microbial production of the methane may well be the largest contribution in the future, right? So however, the microbial conversion of the hydrocarbons to methane, right? Microbial conversion of the hydrocarbons to methane could dramatically increase the amount of energy recovered, right? So this is all about these are the applications of the biotechnology, right? That's why the answer is the all. Come to the next question. Removal of which chain results in the maturation of pro-insulin to insulin? Actually, by the removing of that C chain, we inhibit the maturation of the insulin, right? So the answer is 2. This is also the line of NCRT. See here, when we talk about that, in mammals, the humans, insulin is synthesized as a prohormone, right? Like a proenzyme. It needs to be processed before it becomes fully mature and a functional hormone, right? So it contains an extra stretch called the C peptide, and when this C peptide is not present here, so the mature insulin, what will happen if it is removed during the maturation into the insulin, right? So that C peptide is not present in the mature insulin. Clear? That means the C peptide is present in the immature form, and after the maturation of insulin, that C peptide chain is removed, right? So the answer is C.